Hey everybody, it's Mr. Moreau with a little help video on our Nipah virus activity today. Okay, so I'm on the student work document, which is posted to Schoology, and I'm going to read through the introduction here, just a few highlights just to help you kind of have a better idea of what we're doing here. So the intro does introduce us to the Nipah virus infection, which an epidemiologist would study to see whether or not it's going to be much of an issue in the human population, if it's going to be an epidemic, if it's going to grow to a pandemic status. So this Nipah virus is a zoonosis. Plural would be zoonoses. Now that is a virus or a disease that will be transmitted from animals to humans, so it can make that jump. That sounds very similar to something that we, of course, are experiencing now with the coronavirus, with COVID-19. Now for the Nipah virus specifically, symptoms are ranging. You can have individuals that are asymptomatic, so they really have no telltale signs of the infection, or they can even have fatal encephalitis, which is swelling of the brain or inflammation of the brain. So your job today is to do parts 1, 2, and 3. We're going to do parts 4 and 5 tomorrow. So part 4 has you watching this video here, and again, you don't have to do that until tomorrow. And then part 5 is going to be answering some questions that sort of bring everything together. Now, part 1 here is on this document, and that's going to be us looking for patterns and making predictions, sort of the first steps that an epidemiologist, somebody who studies infectious diseases in the human population, something that they would be doing as their first steps. So the Nipah virus actually had outbreaks in 2 different locations, one of which was in Malaysia from September 1998 through May of 1999. And that area of Malaysia called Port Dickson, that's the one that we're studying most here. And epidemiologists had recognized that a higher prevalence of the Nipah infection among pig farmers is really where it was, who was most hardly hit by this infectious disease. So what the epidemiologists did is they collected data from 2 groups: patients, and these are people with encephalitis. So these are people that are experiencing symptoms of inflammation of the brain. And then they also have evidence that from lab studies, for example, the blood of these patients does show the antigen, that infectious agent actually is in fact in their blood. That's confirmed with lab studies. They also interviewed some controls. Now these are the people, the individuals, who do not have any evidence of the disease, but they did come from farms that had some cases of the Nipah virus infection. However, these people, these controls, they did not have any reports of encephalitis, so they are asymptomatic, if anything. So table 1 shows you the living arrangements for people in Port Dickson. They found 97 patients and 147 controls. You can see that in the top column of, the top row of table 1. N equals, that's like N for number. So 97 patients and 147 controls. And you can see the breakdown of the patients. 86 of them were pig farmers or owners. 91 of them worked on a pig farm. So you kind of get the idea of perhaps where this infection may have originated. Of the control individuals, you can see the same, a similar breakdown there. Table 2, again, they interviewed patients and controls. It's the same number, 97 patients and 147 controls. And now we're looking at their reports of the percentage of those people in this study who reported illness among other animals on their farm. So we interviewed the patients that were expressing symptoms of Nipah virus, and for example, 64% of them say that their cats are experiencing illness. 19% of them are reporting that bats in the area are experiencing sickness. And the same goes for the control individuals. And then table 3, that would be the percentage of infected people in this study who reported an increase in sick and dying animals on their farm just in general. So up in the top there, table 2, percentage of people in this study who reported illness among other animals. And then table 3, reporting an increase in sick and dying animals on the farm. So it sort of takes it to the next level there. And then based on tables 1, 2, and 3, you're going to answer questions 1, 2, 3, and 4. And that concludes part 1 for you. Okay? The next part of today is part 2, and that is a student reading. Now that is also posted to Schoology, and I'm going to scooch over to that document now. I already have this sent over to Notability here. And here is our Nipah virus student reading. So I've already highlighted this, gone through and sort of figured out the important parts. And I just want to highlight a few of them for you, because I think a lot of this can be either prior knowledge or things that I know that you can figure out on your own. But some of it is a little bit higher level in terms of what I know that we have not talked about yet before. So I just want to make sure this is clear for you. So for the intro here of what do epidemiologists actually do, so they are those individuals that in the healthcare industry that monitor diseases in populations, and they do so by, you can see I've highlighted steps 1, 2, and 3, and that last one has some new terms there. Detecting antibodies to specific viral peptides in the individual's blood using enzyme immunoassay, EIA for short. That's also sometimes called ELISA. You might come across that term in a Google search or something, but I don't think that's in this activity anywhere. So anyway, that third way that they can detect the presence of these diseases in populations, they detect antibodies. So an antibody, you may have heard that before. I have that off to the side there. That's a blood protein that we have in our blood that can detect a foreign substance. So those are those specialized proteins that we make in response to interacting with or being exposed to a new substance that our body, our immune system, considers to be a foreign body, a foreign agent that may or may not be infectious. And then what those antibodies do is that they detect a specific viral peptide. Peptide is a fancy word for a protein. You know, a lot of viruses have a protein capsule on the outside in the individual's blood. And this enzyme immunoassay, this EIA test, is pictured just down here in this figure, okay? So what they can actually do is, you can see in step 1 here, they're placing some of the actual infectious agent in this culture plate, okay? This culture plate, think of it basically like a bunch of mini empty plastic petri dishes. So they can coat the bottom of those culture dishes with the viral antigen. See that in the zoomed in picture over here, way at the bottom in green. That's the viral antigen actually bound to the surface of the plastic plate. And then they would put the individual's blood in there. This is actually detailed in the bottom section here on this paragraph. EIA involves, I'm reading the bottom of the page now, EIA involves coding a cell structure, blah, blah, blah. It goes on to describe exactly how that's actually done. That's probably an area you want to read so it makes a little bit more sense to you. But basically they can put some of the individual's blood mixed in those plastic wells, and what's going to happen is the individual that has antibodies in their blood, those antibodies will bond to the viral antigen. And you can see the kind of upside down Y-shaped thing, that's the antibody, bonding to the green triangle, that's the antigen. So this is the body's natural blood proteins, immune-based proteins, bonding to the viral agent. And then what they also do is mix it with a second layer, and that substance is a specialized antibody enzyme kind of network, a team, a compound. So it kind of looks like a yellowish brown upside down Y bound to that gray looking like C-shaped thing. So that is an antibody bound to an enzyme. And they do this to ensure that the antibody, the green one that is bound to the viral antigen, also connects to the brownish antibody, which is connected to the enzyme. So essentially they've connected to the antigen an antibody, and then to that antibody, another antibody, which is already connected to this gray C-shaped enzyme. Finally, they would introduce another chemical that is the substrate for that enzyme. Remember, the substrate is something that the enzyme acts on. The product is what the enzyme turns the substrate into. So, the product of this enzyme catalyzed reaction is going to be a different color than the original substrate. Okay, so the enzyme turns the substrate into something new, the product, and the product will be of a different color. So this entire liquid, this entire substance will change color if the enzyme is able to complete this reaction. it would only be able to complete this reaction if the enzyme, which is attached to the antibody, which is attached to the other antibody, which is attached to the antigen, is present. So long story short, if the antigen is present, all of these things can connect, and then the enzyme can do this color-changing reaction. So only if the person has the antigen in their blood will the scientist observe a color change in this EIA test. Okay, so that's how they actually go about detecting whether or not a person has that virus that they're interested in. So that's some of the more complex part about the reading here. Scooching ahead, there's also some very important definitions of some key terms here. So I've highlighted them in some different colors there. And two things that I think we've heard a lot more recently are the words epidemic and pandemic. You can see that they're defined there. And they also go on to include terms such as the disease's incidence, prevalence, morbidity, mortality, case fatality ratio, and basic reproductive number. And those are all individually defined and detailed below. So you can see that I've highlighted incidence in blue, and then I highlighted the definition in blue as well, with an explanation of how it's calculated. And then the prevalence highlighted in purple, and then I highlighted the definition in purple. So you can see that the terms that are in that top paragraph on my screen right now are explained in the paragraphs down below. And that's what's going to help you make the calculations back in our student work document. So part 3, after you've done part 2, the reading, part 3 is going to be you practicing calculations and making claims. So here's some vital info that you're going to need. As Nipah virus spread, I'm reading part 3 here, as Nipah virus spread from September 1998 through May 1999, that's the time range, in Malaysia, 265 patients were diagnosed with encephalitis. Those are the symptom experiencing patients. 265 people actually experienced symptoms. 105 of them died. Additionally, 110 individuals were found to have antibodies to Nipah, but did not have any clinical symptoms. So those people did have the virus, but they proved to be asymptomatic. They did not show any symptoms. Of all those infected, 224 were from Port Dickson. That's that area that we were talking about more specifically that was being studied. So that's more of a geographic thing, which has a population, the Port Dickson area has a total population of 97,800 people. Okay, so what I've done is gone through and I looked at the definition of incidence, prevalence, morbidity, mortality, and case fatality ratio. And I did a little color coding system here of what numbers I'm going to need to figure out the actual percentage. So for example, for prevalence, I'm looking at 1B here, prevalence in Port Dickson, Malaysia. I have red divided by green. That would be 224 people from Port Dickson were infected, and then 97,800 is the total population. For the prevalence in Port Dickson, I would do 224 infected people divided by 97,800 total people, because that's what gets me the prevalence, infected people divided by total people. So that's what I've done here to kind of show you how the math works. You can screenshot this right now if you'd like, so you can help kind of get some help with the math using my color-coded system to make it a little easier. In any event, I do ask that you try your hardest to not just copy the numbers directly down into number 1 here because of my color coding system. I'm hoping you use this to kind of check your answers as opposed to reading through the definitions for each of these terms and then figuring out which of the numbers actually fits. Okay, so that's what I'm hoping you do. And then that will be the end of part 3 after you answer question 2, because part 4, watching this video, answering the questions, and then part 5, putting it all together, that's going to be not due until our next day of e-learning. But that is it for today. I hope that clarifies some things. And as always, email me. If you email me during my office hours of 10 to 1, I will respond immediately. But feel free to email me at any time. I just might not get to it instantly. Okay? See you guys.